In this video, we are going to check about rDNA technology. So, what is this rDNA technology? It is the full form is recombinant DNA technology. It is a type of genetic engineering in which we manipulate DNA to create a new combination of genes by combining DNA from different organisms. So, we combine DNA from different various organisms to create a new combination of genes. That is, we call this recombinant DNA technology. So, where we use this technology? What is the purpose of this rDNA technology? It is used in agriculture field, biomedical field, pharmaceutical areas, and basic research. In wherever in lab we go, there will be molecular biology lab or any genetic engineering lab. This rDNA technology is the most basic experiment we do, and in gene therapy it plays a very important role. So, who are discovered this rDNA technology are first Stanley Cohen and Robert Boyer and Paul Berg in 1972. So, these are the three people who found this rDNA technology. How did they find? So, they found antibiotic resistant genes present in a plasmid of a drug resistant E. coli. So, from that only they founded this rDNA technology. So, now let's check out what are the steps involved in this rDNA technology. So the procedure of this rDNA technology is so basic and simple. Here I have listed important 8 points which we have to remember while doing rDNA technology. So the first step is isolation of DNA, gene of interest from a donor organism. Second is cutting DNA. So we use enzymes, restriction enzymes like restriction endonuclease are used to cut this DNA at a specific sites. There will be restriction sites. So this restriction enzymes will cut at restriction sites and create small fragments of this DNA. Third is vector preparation. So the most commonly used vector here is plasmid. It is prepared. So we cut the plasmid with the same restriction enzyme which we use to cut our gene of interest. So there will be sticky ends in both sides which can be ligated afterwards. So fourth step is amplification. So the desired gene and the vector DNA are amplified using polymerase chain reaction. So step is ligation. So we will be joining this vector DNA and our gene of interest together using a ligating enzyme called ligase. Sixth is transformation. The recombinant DNA is introduced into the host cell by bacteria or yeast or any other cells, a process called transformation. The seventh step is selection and cloning. So the host cells are screened to identify those that are successfully incorporated the recombinant DNA. So if rDNA is incorporated into the host cell, means it will produce some pigments and we can identify that these cells are taken the rDNA. Then these selected cells are cloned to produce multiple copies of our rDNA. And the last final step is expression. So the cloned gene is expressed within our host cell, leading to the production of our desired protein. So isolation of DNA from our desired organism to the expression of that protein are the important 8 steps in this rDNA technology. Now coming to the applications of this rDNA technology, it has numerous applications, but here we will just check out the most important or basic 3 applications. First, it is used in medical industries. So to produce therapeutic proteins. For example, to produce insulin, in those days it was very difficult and it was very costly. But nowadays we insert this insulin producing genes into E. coli and we make that bacteria to produce human insulin. So that's we get a cost effective insulin. Second is in agriculture. So development of genetically modified crops is possible by this rDNA technology. For example, BT cotton. It contains a gene from the bacteria Bacillus thuringiensis, which we call as BT. So which produce a toxin harmful to certain pests. So this genetic modification reduces the need for chemical pesticides or fertilizers, making the cotton cultivation more sustainable. This rDNA technology influences production of vaccines also. And finally in research. So in studying gene functions, this rDNA technology is so helpful. Like gene fluorescent protein GFP is originally isolated from jellyfish, is commonly used as a genetic marker, but its fluorescence allows the scientists to visualize and study gene expression or protein localization and cellular process and other cellular metabolism. So these are the main applications of this rDNA technology. Like applications, there are some limitations also for this rDNA technology. So there is a destruction of native species of organisms in the environment due to this genetically modified organism. As these genetically modified organisms are more efficient, it is suppressing the growth of these native organisms. Second is ethical concern for manipulation of individual genetic information. So we can't be easily manipulating others' genetic information. So there are some ethical issues there. And the safety of the modified food and medicines using in the rDNA technology is still debatable. Like safety purpose is also one of the limitation in this rDNA technology. Hope at last in this video we learned something basics about this rDNA technology, like what is that and why it is being used, who discovered this rDNA technology and what are the procedures, what is its application, limitations. So this kind of basic information we learned in this video. So hope it was helpful. That's all.